Okay, so now we're recording. So I'm going to start with the wild type MDH1 splice variant 3 and just double click it. When it opens up, go to map. When you go to the map, it might look like this because this show alignments was open. So just close show alignments. Okay. The only difference is this straightens out or flattens out and makes it a linear version. I like looking at the circular map, and it also shows some things later on here, some sequences that were popped in there. So just get rid of this show alignments. So here you can see it's PET28A, human MDH1, that's the cytosolic version, splice variant 1 plus TAV. So that's the wild type plus TAV. I'll show you in a minute what it means. All right. And so in here is the wild type form. If it's a mutant, it'll say like S24D or something after this, and it would say it here too. Um, Later on, remember this for later, there's a T7 promoter. When we do DNA sequencing, we have a T7 promoter and then we have a T7 terminator or reverse primer. This primer reads into the gene from the C terminus or the end of the protein, and this primer, when we do sequencing, reads from the N terminus or the start of the protein end. This protein, if I hover over this and just let it sit, notice it comes up and it says it's HMDH1B3. It starts at 5,071 base pairs and goes to 6,174 base pairs for a total of 1,104 total base pairs. So the number one base pair is 5071. That's important to remember. This thing is 367 aminos long. When we do sequencing, you'll hear me say most of these sequences can go more than halfway through, but not all the way, all the time through the read. So if you're looking at a protein that's on the more carboxy end or towards the terminus of the protein, you want to use the primer reading this way. If you're using something that's halfway through, again, it says we've got 364 amino acids. So roughly if 0 to 150 amino acids, you want to use this promoter. From 150 to 360 or so amino acids, you want to start using this promoter, but that's for later. Just kind of introducing you to how this thing works. And you can see over here, it's kanamycin resistance. Okay, so I'm going to click on this because when I do something when this whole thing's highlighted, it jumps to that sequence. Okay, so if I hit sequence with the gene MDH highlighted, it goes right to the sequence. All right, if I go back to the map and click away from that and then hit sequence, it starts way up here, right? It just, or the last place I was, but it may be, this is a big plasmid, so it might be up there, might be there, so you don't have to scroll around to find it by just literally going map, clicking and highlighting this region of the plasmid, hitting sequence, it'll jump right there, okay? So what is this? Here you see ATG, that's our start codon. Click on it again, gives us that same information, 5071. If I hover over this ATG 5071, 5072, 5073, right? So there's our base pairs for the first start of this methionine, which is ATG. Okay. Sometimes the numberings are off a little bit because sometimes we used to not include the methionine because as soon as the protein is made, the cell removes the start methionine. So really in the mature protein, the first amino acid is this glycine right here. Notice it says in the second position because it didn't ignore the starting methionine, which in a mature protein is cleaved off. So all our numbering is now using and including and keeping the numbering system for the methionine, even though in the mature protein, it's not on the protein. So like in Anastasia, we used to number this with because you were using pi mole and pi mole didn't show the methionine because it wasn't on the actual protein in the pi mole structure. So you always have to look. All right. So let's say I'm going to scroll down here. And what's nice here, if I click on this valine right here, I hold it on there. It says valine. It's a 28th amino acid. And it shows you here's the codon for valine, GTG. Here's a T, that's a threonine. I click on that, ACT. These are the codons for bacteria to make the protein sequence that we see here. Okay. And also you can see 25, it happens to be an arginine, the 25th, there's the 26th, it's a valine, and so on. So let's say this serine right here, serine 21, I want to make this serine into an aspartic acid. Okay. That means I want to change TCT to GAT, or there's a couple of different codons that work for bacteria. All right. So how do we do that? All right. So this is the, oh, wait a minute. I lied. I'm going to go back a little bit. So this is the beginning of the protein here with a methionine ATG, first amino acid. That's the amino terminus. So it's the nitrogen is on the peptide backbone. As we go through this, we get to this green section. So we go to SALE, it's a sale. And then we have 7 amino acids that make up what's called the TEV region. If we wanted to get rid of this histidine protein, part of the protein, the histidine tags, 6 histidines, then the stop codon. If we wanted to get rid of this histidine, we would take a protein called TEV and it would cut this site right off away from the rest of the MDH. 99% of the time, we don't care about it. It doesn't affect the protein. We leave it on there. But if we ever wanted to get rid of the histidine, that's what we do. Colby, Max, and maybe later some of you other folks, you may end up wanting to do this. Okay, get rid of that TEV or histidine by using this TEV site. So there's our whole protein. Okay, so if I go back to the map and I select the whole plasmid by just doing all select, right? I can also just do, I think, edit. Where is it? Copy map. No, I want to do all. There we go. All. Okay. And then I want to go to file or edit. There we go. Edit. I want to copy the top strands. Okay. So there's a top and a bottom strand. And go to molecular tech, genetics, and understand about reading frame and all that. But for now, copy top strand. Bang. Now we have in our buffer the entire sequence for the entire plasmid. Where does our gene start? On base pair 5071. Okay, you need to remember that or write it down. It's not always 5071. So if you're doing MDH2, you have to hover and say, what's my first base pair in the reading frame for MDH2? Okay, so that's step one. Then we need to go to NEB. Oops. It jumped right to it because I was just there. So let's just do neb.com. So New England Biomedicine. Okay, here I'm just going to type site-directed mutagenesis. That'll get you there. The name of their kit is Q5 site-directed mutagenesis. Click on that. All right, and then it'll give you all the products. Let's just check this out for a second. So I'm going to click on Q5 site-directed mutagenesis, and for 10 mutants, it's $233. That doesn't count the cost of the primers, nor the cost of purifying the DNA, and then the cost of sending it out for sequencing. Science be expensive. Okay. But when I clicked on that, I came to this page and you get protocols, backgrounds, it gets FAQs and all that stuff if you're curious. It even has a little video. Okay. Today is not the day to show you how site-directed mutagenesis works. That'll be next week. This week, I want you to design primers. We'll worry about how we use the primers after the primers get here. All good? Verbally shake your head.
Yes.
Thank you. Oh, it's reassuring to know you're still there because I can't see anybody. All right, so click to NEB base changer. Boom. That's the website. Here's where people get kind of mucked up. Remember, in SnapGene, I selected the entire plasma by doing copy all. And I copied the top strand. When I paste it, there's the entire plasmid, all 6,000 or whatever base pairs. Now, is this the first amino acid or a base pair for our starts? No, it's somewhere in the middle. 500 or 5,071, wherever that is, somewhere in here. Okay. So don't worry about any of this other stuff. That's for other things. Look, there's 6,000. So it's 6.3 kilobases. That's what the map said. Good. So what are we doing? We're just doing a single amino acid AA point mutation. I'm not sure what that is. You can click on this and it'll tell you stuff. This is going to say here's a first base pair. So TGG codes for a tryptophan. That's what W is. But our codon ORF is open reading frame. That's where that methionine is over at, and you got to tell it 5071. Notice as soon as I do that, it whipped all the way down to 5071 and said, okay, here's your 571st, 72nd, 73rd. Those first three base pairs code for methionine. Okay, the next three code for a glycine. The next three code for an arginine, another arginine. Okay, how do you know for sure that's right? You don't take anything for granted ever, ever. Okay, so then you're going to go back to this map, click on MDH, and hit sequence. MGRR. Okay, we're in good shape. So we are in the right reading frame. Always double check. Okay. Now remember, we wanted to take serine, the 21st serine, methionine GRR 1, 2, 19, 20, 21 is serine, and we want to convert that to an aspartic acid or a D. Okay. So S21 tells us the 21st amino acid in the wild type proteins is serine, and we want to change it to a D. So S21D is our nomenclature. So if I go back to NEB, I've already told it what the base pair is. Look what happens if I do 570. The first, it says it 570 instead of 571. It's not ATG, it's CAT, which is a his. See the importance of making sure you start with the right base pair and double checking. This should always be clicked. The codon usage is for bacteria. We'll explain that later. Here again, I'm showing how to make your primers. And so here we said we want to go S21, the 21st amino acid is an S, and we want to make it a D. Okay. You can ignore all these other things. Those are for other programs and other kits that they have. Use the standard NT minimum and maximum primer length and TMs that they have already set for this kit. Then just literally hit build primers. Okay. Notice they highlight the reading frame. Okay. So I'm going to go back to SnapGene. Going to find that 21st serine. Notice it's DDKSEPI. Is that what I'm getting in the mutation that I asked for? Right? So in yellow here, there's a DDK, not an S, it's a D. Hey, EPI. Okay, so this highlighted yellow is the right reading frame. It's going to give us DDK, now a D instead of an S, EPI. So we're gold. Notice in capital letters, this is the normal wild type sequence, and GAT for the top strand or the 5 to 3 prime strand is the mutation we're going to make. So we're going to change what was a CTA to a GAT. Everybody copacetic on that? Please say amen.
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 Bunch of boring Lutherans. All right, I want Baptists. All right, anyway. Um, so we're good to go there. So what you, and also notice in green, they show one strand and then they show the other strand. And that means we have two primers, one in red, one in blue. Again, I don't want to slow down things so you understand the concepts. We'll do that next week. We'll talk about how the site-directed mutagenesis works. And Isabel and Pat will roll their eyes and say, yes, Jesus God, we've already done this. All right. So what you need to do is here it says replace TCT between bases 5130 and 5133 with GAT. So it's S21D. Awesome. And then there's two primers. The top one is the forward primer in red with the new base pairs. Its length is 24. Its TM is 56. The reverse primer is this in blue. It's shorter at 18 and it has a different TM. And this 57 is what we need to do when we do PCR with it. It's going to split the difference between the TMs if they're far apart from each other. All right. So what you need to do is you need to copy all of this. The first person that screenshots this or turns this into a PDF and sends it to me gets the Patrick treatment. Right, Patrick?
Yeah, do not do that.
Do not do that because I don't want to have to read this and type it into IDT webpage, right? I want to be able to copy paste. So what you'll do is you'll just highlight it, copy it, go to a Word document, paste it. Whammo, right? It's a little bit big. Let's decrease it a little bit, right? But I can see that here, right? I can actually change the orientation and stuff, or I can do a few things to this. I can just move this over. There it is. So now what I will do is I will copy paste this copy. I will then go to a new web page, IDT, DNA, and of course, accept all cookies because you already know what the hell I'm doing anyway. So I'm going to go to oligos and reagents. Oh, they changed. There we go. Custom DNA, DNA oligos. And I'm going to order, let's see. Yep, here we go. I'm going to order now. And in this case, I'm only going to order 2 primaries. I'm going to call this MDH1V3S, what was it, 21D, right? And then I'll put forward. Then down here, I'll paste because you didn't make me type it. And I'll tell it lab ready, so it comes in solution, leave everything as is. And then I'll do the second one, and then I can go order it. Make sense? So I need to stop the recording.